Hello everyone, welcome back to my channel Competitive Botany. In this video, we are going to learn about the important terms of genetics. If you not yet subscribed my channel, please subscribe the channel for more videos and notifications. So let's see the terms one by one. The first term is gene. Gene is an inherited character that determines a biological characteristic of an organism is called as gene. So, look at the picture carefully. So, gene is a unit of chromosome that determines a specific trait or character in an organism. For example, so it is nothing but unit of DNA. Remember, that gene determines a person's eye color. So, for example, I am telling different eye colors observed in humans, like generally most of the people have black eye color, whereas some people have brown color eyes. So, that character is determined by the unit of DNA, nothing but gene. Next term, factor. Mendel called the determining agent responsible for the trait is called as factor. Actually, so Mendel termed it is as a factor. Actually, it is alternative name for the gene. Mendel called the determining agent for a character as a factor. Next, alleles. Each gene has two alternative forms known as alleles. So, they are present at the same locus on homologous chromosomes. So, look at the picture carefully. For example, in pea plants, the plant height determined by a gene that consists two alternative forms such as capital T and small t. Capital T determines tallness, small t determines dwarfness. Okay, so these two alternative forms of a gene are called as alleles. So among these, one is dominant over the other. So they are present on two homologous chromosomes in the same locus. Look at the picture. So these two, capital T and small t, both are the alleles. One is dominant over the other. Next, trait. Actually, trait is an inherited character of an organism. So, it is generally determined characteristic of an organism. For example, remember, trait is nothing but characteristics of an organism. Next, homozygous. When a gene pair in an organism contains two identical alleles, capital T or small t, the organism is considered as homozygous. The gene pair is called as homozygote and the condition is referred as homozygosity. So, look at the pictures carefully. So, if an individual have two copies of same alleles, look at the picture. So, these are the two copies of these are nothing but alleles. They are, if they are similar or dissimilar or maybe dissimilar. Here we are observing the similar alleles are present on homologous chromosomes. So, actually what is meant by homozygous? If an individual have two copies of the same allele like capital T or small t, such individual is called homozygous. Actually, they are present on two homologous chromosomes on the same locus. Remember, it should possess a pair of identical alleles. That condition is referred as homozygosity. Next one. Heterozygous. When two different alleles capital T, small t are present in a single gene pair, that organism is considered as heterozygous. That gene pair is called as heterozygote and the condition is called as heterozygosity. So look at the picture. Here two different alleles are present on two homologous chromosomes. One homologous chromosome containing capital T, other having the small t. Okay. So when an individual has two different alleles of the same gene, it is referred to as heterozygous, which is also called as hybrid. Remember, it should possess a pair of different alleles. That condition is referred as heterozygosity. Next one, dominant allele. An allele that is always expressed when present regardless of whether the organism is homozygous or heterozygous for the gene. So look at the picture. If the allele is present in homozygous condition, first one, or heterozygous condition, it will definitely expressed. Here there are two conditions observed that are present in organisms. One is homozygous condition, other is the heterozygous condition. So in both the cases, the plant expresses the tallness. Okay. So T represents the tallness in pea plants, which is dominant over the dwarfness. You should remember. So that allele is referred as dominant allele. So in the first diagram it shows homozygous condition and the second diagram showing the heterozygous condition. In both the conditions it will express its characteristic of tallness because it is a dominant allele. Next, recessive allele. An allele that is only expressed when the organism is homozygous for that allele and not expressed when heterozygous. Look at the diagram carefully. When it is paired with a dominant allele, it will not express. It is expressed in the homozygous condition only. Such allele is called as recessive. So here you have to observe the homologous chromosomes having two small t's. In such case only the dwarfness will expresses. Okay, so that's why this is called as a recessive allele. Next one, phenotype. Phenotype is nothing but set of observable characteristics of an organism resulting from the interaction of its genotype with the environment. Simply, so here you are seeing two diagrams of a pea plant. One is tall plant, second one is dwarf plant. Okay, so set of observable characteristics of an individual resulting from the interaction of its genotype with the environment. So next, physical appearance, nothing but, okay. So simply it is determined as physical appearance of a trait. Physical appearance of a trait. Next, genotype. The genetic makeup of an individual is called genotype. Simply it is determined as the genetic makeup of a trait. Genetic makeup nothing but genes. Okay, so they are nothing but genotype. Next one, monohybrid cross. A cross between two parents differing in a single pair of contrasting characters is known as monohybrid cross. So look at the picture. Here one flower showing red color, other flower showing white color characteristic. So they are differing or they are contrasting in single character. They are differing in a single pair of contrasting, they are having a single pair of contrasting characters. So such cross, both the parents are differ in single pair of contrasting characters. Such a cross is nothing but monohybrid cross. Next one, dihybrid cross. So a cross between two parents differing in two pairs of contrasting characters known as dihybrid cross. Known as dihybrid cross. Next one. Monohybrid phenotypic ratio. When F1 plants of monohybrid cross were allowed to self-pollination. So generally they were allowed to self-pollination. So F1 hybrids always allowed to self-pollinate. Both tall and dwarf plant appeared in the F2 generation in the ratio of 3:1. That ratio is known as monohybrid phenotypic ratio. So 3 representing the tall plants, 1 is a dwarf plant here. Okay, you have to remember. Next question, next term, monohybrid genotypic ratio. A genotypic ratio is a numerical comparison of the allele combinations the offspring will have, such as homozygous dominant, heterozygous or homozygous recessive, will appear in 1:2:1 ratio in F2 generation. This ratio is known as monohybrid genotypic ratio. So, one is dominant, that means capital T. That means one plant obtained as dominant, pure dominant. Second one, so 2. 1:2:2 means nothing but heterozygous. 2 plants obtained in the heterozygous condition. Again, one plant obtained as a homozygous recessive. So 1:2:1 ratio of F2 generation of monohybrid cross is nothing but monohybrid genotypic ratio. Next one, emasculation. So anthers were removed from the female parent before their dehiscence to prevent self-pollination. Okay, this is nothing but emasculation. Removal of anthers before pollination to prevent pollination is nothing but emasculation. Next one, hybridization or crossing. So pollen from the male parent, selected male parent, is dusted on the feathery stigmas of emasculated female flowers. So that is nothing but hybridization. Then what happened? Then fertilization occurs between the two parents, that is called crossing. So here you have to see the diagram. So first one is male plant and other is the female. Second one is the female. So the pollen from the first parent is dusted on the second plant stigma for the fertilization purpose. This is nothing but Next one, parent generation. The generation that supplies gametes to the filial generation is called parent generation. Here you have seen tall parent and short parent. So the generation that supplies gametes to the filial generation. Filial generation is nothing but hybrids. F1 hybrid, and then they are generally allowed to self-pollination, and then they will give rise to F2 generation. Next one, filial generation. The generation that receives gametes to the filial generation is called filial generation. As I told you before, F1 hybrid, that is nothing but filial generation, first filial generation. Next one, first filial generation. The population obtained as a result of crossing the parents, P1, is called F1 generation. When the two contrasting characters when the two contrasting parents are crossed together, they will give rise to a hybrid. That hybrid is nothing but F1 generation. F2 generation. F1 plants generally allowed to self-pollination. They produce F2 generation. Similarly, F3, F4, etc. generations can also be obtained. F1 plants are always allowed to self-pollination to give rise F2 generation, and then F3 and also F4 etc. generations can also be obtained. Next one, homologous chromosomes. Homologous chromosomes, they are same in morphology and genetic constitution. Okay, generally one set in animals and humans also, one set of chromosomes comes from the father and other comes from the mother. Next one, recombination. Exchange of genetic material between chromosomes is nothing but recombination. Okay, so here see the picture carefully. So the chromatin material is exchanged between these two places. Okay, so look at the picture carefully, that is nothing but recombination. Next, crossing over. The breaking and rejoining of homologous non-sister chromatids during early prophase 1 of meiosis resulting in recombination. Okay, look at the picture here also. So, the chromatin material is exchanged between the two non-sister chromatids of a homologous chromosome. That is nothing but crossing over. It generally happened in the prophase 1. Synopsis. The pairing of homologous chromosomes during prophase 1 of meiosis. Synopsis nothing but the pairing of homologous chromosomes. Look at the picture. So for our convenience, they are labeled with colors. So blue chromosome and red chromosome. So they are overlapped one another. Look at the picture. That is nothing but the pairing of homologous chromosomes. Okay, they are showing chiasma like structures. So at the place of chiasma, nothing but X-shaped configuration, there should be crossing over should occur. Next, Punnett board or Punnett square. Punnett board or Punnett square. So the Punnett square is a square diagram that is used to predict the genotypes of a particular cross or breeding experiment. Okay.